Bismillahirrahmanirrahim. Assalamu alaikum. Bismillahirrahmanirrahim. Assalamu alaikum. I'm Dr. Shagufta Perveen from Department of Botany, Government College University, Faisalabad. The title of my presentation is benzyl aminopurine, moringa leaf extract, and ascorbic acid induced drought stress tolerance in the benzyl aminopurine BAP. That is an important plant growth regulator that affects cell division, chlorophyll biosynthesis, photosynthetic efficiency, and nutrients uptake. BAP has very important role in the plant growth and development. For example, different studies show that when BAP is applied exogenously as foliar spray or as priming agent, it can increase photosynthetic rate, yield, and different metabolites under different levels of stress. For example, in cotton, under 50% reduced irrigations, foliar application of 5 micromolar BAP can increase photosynthetic rate under 175 and 50% field capacity. BAP at the rate of 50 milligrams per liter can increase crop yield in maize under 140% field capacity. BAP at the rate of 50 ml per pot can increase ascorbic acid and total phenolic contents in maize. Moringa oleifera. Moringa oleifera is a fast-growing drought-resistant tree that belongs to family Moringaceae. The leaf extract of Moringa oleifera is rich in mineral nutrients, growth hormones, vitamins, and antioxidants. Various phytohormones include auxin, gibberellins, Z-trim type cytokinins. MLE increase seedling establishment, plant growth, eventually production in the field crops. It increases leaf area, plant height, biomass, and chlorophyll contents in crops. Ascorbic acid, that is also known as vitamin C, can detoxify reactive oxygen species and improve growth of plants under different abiotic stresses. It functions as major redox buffer, regulate different metabolic processes like photosynthesis, hormone biosynthesis, and regeneration of other antioxidants. Foliar application or as priming agent in both ways. Exogenous application of ascorbic acid is effective in increasing the germination rate, shoot and root length, shoot and root dry rate under different levels of growth stress. For example, under 114-50% field capacity. Ascorbic acid, when used as priming agent, can increase growth of Brassica napus and increase yielding pea and improve growth in sun. Pea, that is commonly known as mutton, is an important member of family Leguminosae. It is the second most important food crop that is extensively grown in temperate regions. It is the growth season of the tea is October to November. Tea is rich in proteins, carbohydrates, vitamin C, magnesium, and phosphorus. When used in 100 grams edible quantity. Drought stress. Water is one of the essential factors for the sustainability of life of all living organisms, including plants. Plant development and yield is influenced by direct or indirect effects of climate change. It has been reported that annually 55 million people worldwide are affected by water deficit stress. Water deficit stress can lead to 50 to 70% crop yield reduction globally, according to a survey of World Health Organization. It is estimated that by 2030, about 700 million people, that are about 40% of the global population, will be at the risk due to water stress. Effects of drought stress on plants. Drought stress brings about different morphological, physiological, and biochemical changes. These morphological changes include increase in the number of leaves, leaf area, increase in root length, leaf aging, changes in season stage, Various physiological changes include transpiration, stomatal activity, photosynthesis, osmotic balance, water transmission, and relief relative water content. Biochemical changes includes modulation in the antioxidant defense system, photosynthetic pigments, accumulation of compatible solutes that include proline, glycine, butane, hormonal contents, and secondary metabolites. Plants cope with stress by adopting different mechanisms. These mechanisms may be stress escape, avoidance strategy, and tolerance mechanism. Stress escape mechanism is due to the shortening of the life cycle and biodegradable development. Avoidance strategy is the retention of optimum relative water content and high water usage. Plants' water potential is maintained high through a reduction in the stomatal transpiration and the increase of water uptake from well-established sources. Tolerance mechanism, that is the survival mechanism, it includes xeromorphic characteristics such as the presence of hairy leaves and cuticles that may help to maintain high water potential in plant tissues, osmotic adjustment, and solute accumulation. Hypothesis of the current study was to check whether or not the foliar application of DAP in combination of moringa leaf extract MLE and ascorbic acid SASA help in mitigation of drought stress of plants. Objectives were to identify the effect of DAP, MLE, and ascorbic acid on the growth, yield, and physiology of the plants under drought stress, to investigate the pattern of changes in terms of growth and physiological and biochemical parameters of tea plants when foliarly applied with mixture of PAP, MLE, and DEs under drought stress or non-stress conditions. Materials and methods include pot experiment was conducted at the Botanical Garden, Government College, University of Hyderabad. There were two pea cultivars, that is Meteor and P09. There were two drought stress levels, that is 100% field capacity and 60% field capacity. There were 5 foliar treatment levels, that is control, water spray without any treatment, BAP 0.005%, MLE means moringa leaf extract, that is 30%, ascorbic acid, that is 0.005%, and then a combination of BAP, MLE, and ascorbic acid in 1:1:1 ratio. Findings. The results of the current study shows that shoot and root fresh weight significantly decreased under 60% field capacity in both pea cultivars, that is Meteor and P09. Of both pea cultivars, P09 was higher in shoot fresh weight and root fresh weight. Foliar application of BAP, MLE, and ascorbic acid significantly increased fresh weight and root fresh weight in both pea cultivars of various treatments. Mixture means combination of BAP, MLE, and ascorbic acid was more effective in increasing the shoot fresh weight and the root fresh weight in both pea cultivars. Chlorophyll contents, chlorophyll A and B contents significantly decreased under 60% field capacity in both pea cultivars of various treatments, BAP, MLE, and ascorbic acid. The mixture of BAP, MLE, and ascorbic acid was more effective in increasing the chlorophyll P contents in the pea cultivar P09. Melandialdehyde and hydrogen peroxide contents significantly increased under 60% field capacity of both pea cultivars of various treatments. Mixture of both of various treatments, the BAP that is 0.005% BAP, MLE 30%, and ascorbic acid 0.005%. All these three treatments significantly reduced the MDA and in both cultivars, particularly in Meteor. Activities of antioxidant enzymes like superoxide dismutase and peroxidase significantly increased under 60% field capacity in both pea cultivars. Various treatments, BAP, MLE, and ascorbic acid, significantly enhanced the activities of SP and POD in both pea cultivars under stress or post-stress conditions. Catalase activity significantly increased under 60% field capacity and by foliar application of PAP, MLE, and ascorbic acid. Proline content significantly increased in both pea cultivars under 60% field capacity. All treatments significantly enhanced proline contents in both pea cultivars under non-stress and drought stress conditions. However, MLE 30% was more effective in increasing the proline contents in the pea cultivar mixture. In conclusion, the foliar applied VAP, MLE, and ascorbic acid are proved as growth promoters of pea plants under drought stress. These growth promoters alone or in combination can be effective in increasing the enzymatic and non-enzymatic activities of antioxidants under drought oxidative stress of cultivars. While combined application of VAP, ascorbic acid, and MLE was considered as more effective strategy under drought stress on pea plants. Thank you.